It's my pleasure to welcome Dr. Audrey Duarte, who is a professor at the Department of Psychology at UT Austin. She joined UT Austin in 2021 after 13 years as a professor at the Georgia Institute of Technology. Dr. Duarte received her PhD in neurobiology from UC Berkeley, conducted postdoctoral work in cognitive neuroscience at the Medical Research Council in Cambridge, UK. She's highly accomplished, led and contributed to edited books on memory, and served as multiple leadership positions in her department and as on committees in professional associations. As a leading cognitive neuroscientist aiming to understand the neural mechanisms of age-related changes in episodic memory, she uses multiple methods, including EEG, functional MRI, all matched with neurophysiological and neurological patients, neurophysiological assessments. Major aim of her research is to understand the neural changes that underlie age-related decline, a topic that affected a lot of our research efforts. And with, among several interdisciplinary current collaborations, she has particular interest in the cognitive neuroscience of aging in racial ethnic minorities, the psychological factors that race-related stress, depression, and influence memory and underlying brain functions, diverse populations. So please join me in welcoming Audrey.
I realize this is the third time I've been to Dallas in like the last few weeks. I did a college tour with one of my kids, and then Jack, and then here. So thanks for having me. I'm happy to be back here, having a really good time. So today I'm going to talk about things I think are very familiar to a lot of people in this room, which is nice. So I don't have to elaborate on a lot of background, but I'll do a little. So we probably all know this, right? So episodic memory it declines across the adult lifespan. This is, this is in cognitively normal, unimpaired aging. So if we think about episodic memory, we can define it a lot of ways, but I'll define it as long-term memory, personally experienced events like what we had for dinner last night, we were with, and in daily life, there are many real-world examples of episodic failures, right? So forgetting where you parked your car in the lot, not forgetting that you parked it, but where, specifically forgetting someone's name, that happens to me more and more every day, and forgetting important dates. These are all, or just dates, these are all common episodic memory failures that become more common as we age. So a few years, well, more than a few years, yes, some of my colleagues and I, some of us in this room, published this article where we talked about cognitive aging, mechanisms of cognitive aging, and a bit about individual differences. And I really like this graph. This is from the Betula study from Sweden, and it's showing pretty clearly, I think, like the range of cognitive performance across a composite of tests in people from age 35 to 85. And while more of the low performers are in the older range, there are clearly people who are well in the 70s, 80s who are performing as well as people in their 30s, right? So we talked about a lot of variables that might underlie some of these individual differences. And the ones that we've been particularly interested in investigating in my lab are these that we can kind of lump into environment, which is just sort of a broad category. Arguably easier to modify things like sleep, physical activity, mood, and even regulation of stress related or stress more than genetics. So these are the ones that we tend to focus on. I like a few of these. The ones I'll talk about today are sleep and depression, but these are modifiable, right? So we can modify them to some extent with pharmacological treatments, physical activity, therapy, brain stimulation, etc. So I'll talk a little bit about intervention, plans for interventions. So the first part of the talk is going to be about sleep, and I'm going to focus on habitual sleep, so natural sleep rather than sleep deprivation, which I'll touch on a bit. But the big question we're interested in my lab is, does poor habitual sleep in aging negatively impact episodic memory? So, all right, so sleep quantity, just things to look forward to, y'all. I mean, last night I woke up at 1 and I thought it was 6 a.m. Oh my God, here we go, topical. So sleep quantity and quality decline with age. It's pretty well known. So total sleep time decreases, wake after sleep. This is the amount of time spent awake after falling asleep. Something like up to 70% of older people report some sleep disturbances, most commonly insomnia. That's the most common. Okay, so we know from a wealth of literature for many decades, from animal models, humans as well, that sleep is essential for many aspects of health, all aspects of health really, and has an important role in cognition and cognitive decline. So a few years ago, a former student of mine, Emily Hockhead, some of my colleagues, we published this meta-analysis looking at the relationship between individual differences in sleep quality and quantity measured in several ways: polysomnography, self-report, actigraphy, and found that sleep was similarly related to episodic memory performance in young and older adults. And that was that wasn't necessarily what we were going to find, because there are quite a few studies looking at sleep deprivation showing that it seems that older adults may be less sensitive to the effects of sleep deprivation in a study in a lab where you have people code, deprive them or allow them to sleep after, and then test their memory performance. Older adults may sometimes seem less sensitive to that, like not as impaired as young people. But this suggests that a person's kind of natural sleep may be related to memory performance to a similar degree for young people. What we couldn't do in this study is investigate how these relationships are affected by one's racial and ethnic demographics. And why that would be important to investigate is, I'll get to in a minute. Largely what we know about this is from large kind of epidemiological studies. Before I get into that, I want to just make it clear how we think about race and ethnicity in not just my lab, but in the field. So race and ethnicity are proxies for psychosocial, economic, and structural factors. Things like discrimination-related stress, education quality, access to healthcare, quality healthcare, that sort of thing. When we're investigating how race, ethnicity may modify some of these relationships, like sleep memory, for example, we assess these social and structural determinants so we can understand these as contributors, mechanistic factors, moderators of these group differences in cognition. So back to this, what do we know about sleep in racial and ethnic minority populations? So a lot of this is, again, large epidemiological, public health type work. And so this is self-reported sleep quantity. These are just a couple examples, there are many. But here what we're seeing is that short sleep, which is less than 5 hours on average per night, is greater in the Black African American population and some of the other groups here too. I'm highlighting Black African Americans and Mexican Americans because those are the two groups that I study the most in our research, or we study. And there's also over here long sleep, which I'm not going to talk about as much, but I can later, where there does seem to be on average kind of a good sweet spot for sleep. Very long sleep, greater than 9 hours, and very short sleep, less than 5. In the literature, has been associated with worse outcomes, which I can talk more about later. And we can also see these disparities in objectively measured sleep. Actigraphy, if you're not, so actigraphy is a way that you can objectively measure sleep quality with different kinds of wearable devices that monitor movements essentially throughout the 24-hour-long person's wearing it. And here we also see worse sleep, and this is sleep. This is how much time people spend in bed asleep is worse in Black and Hispanic relative to non-Hispanic. So poor sleep in cognitively unimpaired adults, so these are another word for this cognitively normal people, is predictive of Alzheimer's disease diagnosis. So this is from a longitudinal study over a 6-year period where people at time 0 are cognitively unimpaired. And they're divided by actigraphy data into highly fragmented sleepers and minimally fragmented sleepers at baseline. And so 6 years later, we see higher probability of developing Alzheimer's disease and at a faster rate in the highly fragmented sleepers relative to the minimally fragmented. All right, so why that is, there are lots of reasons for that, that are still being studied, but that is this relationship has been shown quite often. So it's also the case that people from these historically minoritized groups have higher prevalence of Alzheimer's disease than non-Hispanic whites. And we all know that the number of people over the age of 60 who are at greater risk for developing Alzheimer's disease is growing substantially. And so will these numbers of people from these different groups. So any malleable factors like sleep are really important for us to better understand how they impact cognition. So a former student of mine, Emily Hockett, who's on the job market, which is excellent, is very interested in this relationship between sleep and episodic memory in people across the adult lifespan from these minoritized groups in particular. And these are some of the questions that we're asking in some of the studies I'll show you here. To what extent does poor sleep quality contribute to the effects of age on episodic memory and the related neural mechanisms? And what's the impact of poor sleep in historically minoritized adults on episodic memory? So this is a procedure that we wrote on a couple of the studies. We have people come in, perform some tests, give them the accelerometer. It's this really attractive watch. Works great, but it looks terrible, so people don't tend to run off with those. But they wear those, and we are able to monitor their sleep quantity and quality using those devices. They perform this paired associate learning task, which I'm assuming people are really familiar with these kinds of tasks in this room, at retrieval, which is during the same day. So in this day 8 visit, that's when they do the memory testing, encoding, and then retrieval subsequently right after. I'll just mention one thing here, which is we test their memory for both intact pairs. So phone candle is intact, even though the positioning switched. And whether it's intact or rearranged, and that intact rearranged will come to play in a little bit. And here's some of the information about the demographics of the groups. This was more of a pilot study. We now have studies with larger samples. So we found, this is sleep actigraphy data over here. So we found kind of what we anticipated, which is in our racial and ethnic minority groups, so we collapsed across the subgroups. We didn't see much difference between the subgroups. I know the samples are small, but we collapsed for the sake of power here. Average sleep time was reduced relative to non-Hispanic whites across age. This is a lifespan sample, adult lifespan. And duration stability. So this is night-to-night stability in sleep duration. That was also reduced in the racial and ethnic relative to non-Hispanic whites. Okay. So over here, this is the relationship, and we find an interaction with age here. This is the relationship between sleep stability and duration and memory performance. So this is deep chronic word pairs. And we find that people who have a lower stability in their night-to-night sleep duration have worse memory performance, particularly with increasing age. So this suggests that this habitual sleep patterns and having inconsistent, night-to-night sleep quantity, and we also see it for quality, like efficiency, may be particularly impairing memory memory performance in. So we've since replicated this kind of pattern, which we're calling, I'll use this term again, double jeopardy pattern. So if you're a poor sleeper in this case and older, impairment seemed greater. But here we're looking at larger samples of Black people who identify as Black African American versus non-Hispanic whites. And here we see that people with poor self-reported sleep, this is from a subjective sleep measure, have worse memory retention over for paired associates over 72-hour delay. That was specifically in our Black participants who were middle-aged. So we're seeing this kind of negative synergistic effect of sleep and in this, sorry. So we're seeing this kind of negative impact of poor sleep in this minoritized group, Black participants, even in middle. So, a question is, why is that? Like, why is this poor sleep more impairing of memory performance in some minoritized participants? There can be many reasons for this, but again, race being a proxy for other factors, psychosocial factors, we've looked at race-related stress being one of those. And there are, this is from a questionnaire called the IRRS, and we ask people to rate these, it's a Likert scale, so you just respond on a scale, endorsements of these statements. This is an old questionnaire, but it's, topical still. You have been subjected to racist jokes by whites, non-Blacks, and positions of authority, and you did not protest for fear they might have held it against you. Another example, you notice that when Black people are killed by the police, the media informs the public of the victim's criminal record, or negative information in their background suggesting they got what they deserve. So people rate these as more, endorse them more strongly. Their sleep is worse, right? That's generally what we're finding. And this is across groups, and you might wonder, well, how could non-Hispanic whites fill out this questionnaire? It's because not everybody does, but some people do, and they can if they are talking about their experiences with like a family member. So we had some, for example, non-Hispanic white adults who had children who were biracial. So that's that. Okay, so this is all sort of negative, right? Stuff, but we've recently been exploring, factors that they also differ between racial ethnic groups, but are potentially positive buffering factors. And one of those is religiosity. So people from Hispanic groups and Black African American groups tend to higher on self-reported religiosity, whether it be public or private. And these are, you ask people to answer these questions like, how often do you attend church, or in my life I experience the presence of the divine. And what we see is that in this study, that people who are higher on religiosity, whether it be public or private, it didn't matter. The negative impact of race-related stress on sleep quality is attendance. So that's interesting. And what that is about religiosity, we don't know from this study, but there is other work looking at the positive attributes of religiosity, and one of them being emotional regulation. So that's something we are exploring in work along with religiosity, is emotional regulation. And social support is another. All right, so we've also been investigating the underlying neural mechanisms of these relationships between poor sleep quality and episodic memory in adults. And so probably a lot of you know, but neurocomputational theories of memory suggest that successful episodic memory retrieval is somewhat dependent upon the reinstatement of encoding related neural activity and associated cognitive operation. And one way we can measure that reinstatement is with a method called encoding retrieval similarity analyses, where we're looking at the overlap in neural activity between coding of an event and the subsequent retrieval of that event. Several studies have shown that ERS is reduced with age, and higher levels of encoding retrieval similarity predicts better episodic performance. But we really don't know much about the impact of these sleep disparities that I've been talking about on these neural indices, ERS, of successful memory performance. So that is something we investigated in this recent study. Another one of my former students, Sarush, contributed to this analysis. And there's a lot to this, but I'll just kind of give you a very brief overview of it. So we used EEG for this study. That's one, you could also use fMRI, but we used EEG. And we measured EEG while people were performing the encoding and the retrieval of these paired associated learning tasks. We ended up sort of dividing the electrodes into these 4 ROIs, scalp ROIs. And then for every event, we looked at neural activity measured in the time frequency domain of encoding and retrieval. And we looked essentially at the correlation between every time window during encoding, which is like a 250 millisecond window, and set every window of retrieval for that same event. So we're looking at the correlations between encoding and retrieval in time frequency space. So that is the method that we used here, and I think I can advance it. All right, so over here on the left, and this is in the same participants that I showed you earlier for this adult lifespan sample. But here what we're finding is for those intact pairs. So remember that some of the pairs are intact and some of them are rearranged. And so to be accurate for an intact pair, you just need to say, yes, that's intact. I've seen that together. Handle fine. But if it's rearranged, what we think is that people would need to recall what they previously encoded. So if you have a rearranged pair, each of those items in the pair was associated with another item. So in order to correctly reject this rearranged pair, you would need to recall to reject, right? So what we're seeing here is that people with lower sleep duration stability have higher encoding retrieval similarity, ERS, and that was particularly true for the racial and ethnic minority across age. But over here, this is for rearranged pair encoding retrieval similarity. We see across groups that people with lower sleep stability show a lower ERS. So that was sort of a pattern we anticipated we would see: worse sleep, lower, neural index of episodic reinstatement. This one surprised us a little bit until we started thinking more about, like, well, for intact pairs, one could use this process called familiarity to endorse this pair as, yes, I've seen that before. And so what we are speculating, because we didn't test this directly in the study, is that people from racial and ethnic minority groups who have poor sleep stability may rely to some extent on this familiarity-based sort of recognition to endorse these pairs of things.
Audrey, so on a point of information, how do you do encoding retrieval similarity for rearranged pairs?
So we,
different items of encoding.
That's right. So we have one pair at retrieval, which is rearranged, and then we had the 2 pairs, corresponding pairs for that rearranged pair. And we looked at the overlap for those 2 pairs, averaged them together to compare with the
Yeah, I'm not sure I would call that reinstatement. What is it that's being reinstated in a rearranged pair? Thinking it's recorded. Yeah.
That's what we think. Yes. And this is the difference between correct rejections and false alarms. So we're looking at, so this is event specific. So this is, gosh, this is a complicated analysis, but we have event specific reinstatement relative to great above and beyond the reinstatement for all other rearranged pairs. And then on top of that, we're looking at greater ERS for those trials where they correctly reject this one as, this is rearranged, relative to false alarms when they falsely endorse that as being. So in that prior study, there was no delay between encoding and retrieval, just encoding, then retrieval. And we looked at sleep as sort of a individual differences variable, show those relationships, but we couldn't assess how sleep during consolidation between encoding and retrieval was associated with performance or neural activity. And the reason that would be interesting and important to do is because there's a wealth of literature showing that, episodic memory is somewhat sleep dependent, where if you, classic studies are deprive people of sleep between encoding and retrieval or animals of sleep, then episodic memory is reduced, performance is reduced. And so we wanted to assess this kind of sleep dependent consolidation using habitual sleep instead of deprivation. So we have a different kind of a task here. It's another sort of paired associated learning task with objects and scenes. And people perform this encoding, immediate retrieval, so we can assess retention. They sleep for 72 hours, come back to the lab, and then do delayed retrieval. We ask them whether this is the same pair. This was different. So this would be a different pair, or it's new. So specifically for the mismatched pairs, again, where we think people have to engage in this kind of recall to reject mechanism, right? This is different because I remember I think that celery was on the playground. That's how I know it's not associated with the dining room. We found that this greater, people who have higher sleep duration during that retention period, also sleep efficiency, which we measured, is associated with a stronger ERS signal for these mismatch pairs. And that was across groups. We also looked at ERPs, about related potentials, and one in particular that was associated with sleep across participants was the late posterior negative. So the LPN has been associated with processes during retrieval associated with things like episodic reconstruction, which again kind of fits this idea that if you, and it negative, it's the LPN because it's more negative going for correct rejections false alarms. So that's why the slopes are negative. So again, across young and old in this study, a stronger, better sleep is associated with a stronger LPN. Again, kind of suggesting collectively that perhaps sleep quality, one's individual differences in sleep quality and quantity are predictive of episodic memory performance and retention of sleep field delay. Okay. So we've used actigraphy, we've used self-report too. Actigraphy is great for accessibility. It's relatively unobtrusive, right? Just giving people a watch usually, and they can sleep at home. And but you know, it's not perfect because it can overestimate total sleep time. So if a person is lying still but not sleeping, that can be that can register as sleep. We know from some this is more self-report, but racial and ethnic minorities in some studies and older adults sometimes overestimate their sleep quality, which is why self-report is you know, not the greatest always. And importantly, we can't assess the neural architecture of sleep. And that's important. That's the gold standard of sleep assessment is polysomnography, measuring EEG, where we can look at these different sleep stages and sleep features. And these microarchitecture features have been implicated in you know, episodic consolidation and may be affected by, are affected by, for example, Alzheimer's disease pathology. So they can also be targets of therapy. So it's important to be able to also measure sleep architecture. And if you aren't familiar with this, the typical way to do a sleep study, like a traditional one, is polysomnography. And these are just some examples. This is, you know, have people come into the lab. This would be like not old school, but like a traditional setup. Not the greatest, right? It's a little awkward to sleep this way in an unfamiliar lab setting. This is a mobile system, which is better because somebody can kind of move around and go home, but still not, it's a little bit obtrusive. But with a collaborator of mine, Dr. Han Yo from Georgia Tech, we've developed this sleep monitoring patch system. So this is one of our projects who's applying it at home. And it's adhesive, it's like Band-Aid-like sort of texture. It has gold printed electrodes built into it. This is the battery. It also contains an SD card and Bluetooth. So people can take these, or we can mail them to subjects. They apply them at home. And we've recently completed a home deployment of this patch. There's a tablet application, so here he is using it. And we get really great signal from it. So we're pretty happy with this. So we can measure reliably the different sleep stages. So this is our device versus traditional polysomnography from our validation study. And then this is data we've collected from subjects' homes. This is a way of just kind of measuring in the frequency domain, the different stages of sleep. And these are called hypnograms. So basically the black and the red just kind of are showing a matching between our device and polysomnography. And this is showing percent agreement between scoring of the two different systems. So which is good and on par with many other studies. So we're quite, and the home deployment has gone well too. So we're writing that up now, but our goals with this wearable system are to overcome existing limitations in sleep monitoring, which I already kind of mentioned. They're low cost, the environmental impact is low. We can recycle all the components, apart from the electrodes, which we cost, but we recycle all the rest of it. We can mail these to participants, so they don't even have to come in, because accessibility and ability to sort of take time off of work for a lot of people, or even have transportation to get to us, is not always a given. So we are starting to enroll people and then mail them these devices. We're able to identify features of sleep architecture, so we can identify spindles, slow oscillations, and all of the features that have been linked to memory and Alzheimer's disease in the literature. And where we would like to go with this is to be able to identify targets for future personalized brain stimulation, cognitive behavioral therapy, or other kinds of things. So I think it has a lot of potential. They can also be used to record wake EEG, which we're kind of tinkering around with that idea right now too. I'm pretty excited. Sure, but if you all do, typically just kind of power through, or just if you want to ask questions, feel free. I'm happy to not hear my own voice for trade or whatever. So another factor that we've been really interested in is depression, and depression has a really important association with memory. And what we know relatively little about are the consequences of depression in older age. So now, older people, I'm thinking here like 65 and older, are less likely than younger people to be diagnosed with major depression. So that is true. But older people tend to have a higher prevalence of subthreshold depression. So this is exactly what it sounds like, which is symptoms that fall below diagnostic criteria. It's also true that older people who present with depressive symptoms, they tend to endorse, and there can be various reasons for this, which we can talk about in this room if you like. They tend to present with more somatic and cognitive than affective symptoms. So malaise, body pains, kind of nondescript body pains, rather than like sadness, melancholy, and despair. And that could contribute to underdiagnosis, recognition by themselves or physicians of the depression. There are quite a few studies showing that older people who do have higher levels of depressive symptoms are at a greater risk of cognitive decline. But we know very little about the mechanisms of cognitive impairment and depression across adults. Most of the work has really been done in younger people. So this is this is from Lisa Barnes some time ago, but looking at people over a period of, I don't remember how many years this was, 6 years. But so here they're looking at depression symptom score, and this is the CESD, so it goes to like 40. So what this is indicating is even relatively low levels of depression, and this is mild to the moderate range, are associated with a higher risk of developing mild cognitive impairment as follow-up. Right? And everybody here is, at baseline. So I don't know how many of you are familiar with these different kinds of depression questionnaires, but this is one that we use a lot. We like the CESDR because it has subclusters, somatic, it has some anhedonia questions, and it's been validated in racial ethnic minority groups, so it's a good measure. But you ask people to rate these different, endorse these different statements. I felt depressed, I could not get going, I lost interest in my usual activities. So higher score, higher level of depression. So in recent meta-analyses from my lab and from my collaborator Vanetta Dotson's lab at Georgia State, we estimated the magnitude of depression severity, including subthreshold depression, on episodic memory and executive functioning. And this is a meta-analysis, so it's multiple kinds of methods, approaches to assessing episodic memory and executive functioning. But what we found is kind of a similar kind of negative slope here, which indicates that the negative impact of depression level on cognitive functioning, episodic memory, and executive functioning is exacerbated by it. So we have been calling this a double jeopardy. A older, higher level depression, worse cognition. And this is cross-sectional data. I'm just going to point out one thing here, which is that in the episodic memory domain, there's been a lot of research looking at different kinds of valence of events, and the typical pattern is that depression is associated with worse memory for neutral events. But for negative events, those studies that have done this, which are mostly in younger people, show a relative sparing. I'm going to talk about that in data. So we've developed this executive dysfunction theory of double jeopardy effects in episodic memory. And this is based on a few points of rationale. So executive dysfunction is a primary contributor to episodic memory impairments in cognitively unimpaired aging. We've shown that, others have shown this too. You don't always see episodic memory impairments, but they tend to be more noticeable when the tasks are highly demanding of executive functions. Things like inhibition or post retrieval monitoring. That's executive dysfunction is a likely contributor to emotion regulation deficits and major depressive disorder. So I'm going to talk a little bit about emotion regulation in a little bit, but when we regulate our emotions, either suppress our negative affect or reappraise a negative situation as less negative, that requires a lot of executive control. And it's thought that poor emotion regulation in depression is part of that is because of executive dysfunction. We also know that similar prefrontal brain areas largely underlie executive functioning, emotion regulation, and age-related episodic memory. So putting it all together, that's how we have developed this. So in some series of studies from my lab, we wanted to investigate the extent to which depression symptom severity would reduce the ability of adults across the adult lifespan to spontaneously regulate their negative affect. In particular, older people, which would be consistent with this double jeopardy pattern. So previous studies of emotion regulation have really only looked at motion regulation to the stimuli when they're being presented, right? So looking at negative pictures and measuring kind of emotion regulation behaviorally, physiologically, neurally. And there is what we wanted to look at here is spontaneous emotion regulation in anticipation of negative events. So often in the real world, we can anticipate a negative event, right? So we hear screeching tires driving, that you can imagine is something not great might happen after that, right? Or like see dentist appointment, root canal on your calendar, right? So there could be something coming up that you can anticipate and may be able to employ emotion regulation to reappraise that negative situation or suppress it. And so we wanted to determine whether spontaneous emotion regulation of negative events was reduced with level of depression. We also wanted to look at memory, which a lot of studies, emotion regulation studies, haven't looked at the impact of regulation on memory in adults across lifespan. And we wanted to answer this question as to whether neural mechanisms of emotion regulation are altered with depression severity. And this work was done by two of my students. So this is one of the tasks that we've used. We have during encoding cues, they were auditory cues, either tires screeching, and these are all standardized auditory cues that are negative. People rate that as negative. Positive would be like a cash register sound, and neutral is a chime. So they get one of these cues, there's a delay, and there is a picture, and these are standardized negative, these are standardized emotional pictures. And what we're interested in the neural activity, which I'm going to show, is during these catch trials where there is no stimulus here. And so we remove the stimulus from these catch trials, and people they don't know when the cue comes up whether it's a real trial or catch. And that allows us to measure spontaneous emotion regulation without the confound of the stimulus and emotion regulation stuff. And then people subsequently in the same day made these recognition and confidence about this. Okay, so this is behavioral data. Over here on the y-axis, this is negative memory preference. So this is D prime for negative events, subtracting the D prime for neutral events. So everybody is above 0, indicating a negative memory preference. So that's typical in the literature. But what we're seeing here at low levels of depression on that CESD, there is a reduced a negative memory preference, basically in older people. And that's sort of typical in the literature that in older people, we tend to see reduced negativity preferences, sometimes positivity preferences, positivity biases. So that's sort of consistent with that literature. On the other side, at higher levels of depression, we see this crossover. So what we're seeing is in the older, these are simple slopes. Okay, so it's like the upper third standard deviation here. But the higher level of depression is associated with greater memory of like an increased negativity bias, essentially, especially in older people. So that could be, consistent with the idea of, congruence with your negative affect could lead to attentional biases to negative and enhanced memory for negative. What was a little bit surprising to us was this. So for younger people, higher level depression associated with lower memory. At first we thought, well, that's kind of odd, but given that younger people in the higher level depression group, that they also had lower levels of self-rated arousal during the task, because we measured that during the task, we thought perhaps what we're seeing here is some anhedonia, reduced arousal in people with higher levels of depression. We didn't measure anhedonia as like a lack of interest in pleasure, and that is an aspect of depression as well. So we didn't measure anhedonia directly, but we are doing that research now. Yeah.
I have informational questions that might give this some context. I'm curious. So you're saying levels of depression, but you really mean scores on CESD?
Mm-hmm.
Do those people have major depressive disorder?
Some.
And how many? And is that quantified in the analysis?
So some people have a diagnosis.
Okay.
Most do not.
Okay.
There's a range of depression severity from mild to severe, and diagnosis had no impact on it. So, and neither did medication.
And I was wondering, because you look at the subtraction of the negative versus neutral, if you look not at that subtraction, do you see any evidence of like just lower overall?
Lower overall episodic memory?
Yeah.
I don't remember. I want to say no, but I don't remember for sure. That's it, yeah. So they just have percent. I feel like that's not, I think it was specific. Okay, so here, this is the fMRI study. So we're measuring the fMRI activity. Well, we're interested in the catch trial period, the spontaneous, where there may be spontaneous emotion regulation. And these are the ROIs that we use because of their role in emotion kind of regulation in the literature, encoding and visual processing and kind of emotionally salient stimuli. So we used functional connectivity analyses. That's what I'm going to present here. And that's what we're seeing on the y-axis. So this is just one example of the C to C connectivity between our ROI and our connectivity between, in this case, the dorsal medial prefrontal cortex, which has been associated with emotion regulation, top-down control of regions like ventral occipital-temporal complex, amygdala too. And at low levels of depression, self-rated depression, we see typical aging effects, which have been shown in literature where you see this kind of inverse coupling between these regions during emotion regulation in other studies. But here we're seeing this during anticipation of negative event, even though there's no negative event after. And so why do we see that? What could this reflect? It could reflect, we think, anticipatory suppression, perhaps, of perceptual, what you're about to see, and sort of, we don't know, right? Because we're not asking what they're doing, but that was sort of one thought happening. At this end, we're seeing that with increasing depression, this coupling is reduced in older age. So, putting this all together, is what we think is happening is that with increasing age and higher level of depression, there is greater executive dysfunction, right? Less kind of top-down control via these prefrontal regions that are important for emotion regulation, less emotion regulation, and enhanced negative biases. We're testing this like executive dysfunction theory.
Just expand on. So the y-axis is showing the difference in connectivity between negative and neutral files.
Correct. Yeah.
So towards zero, you're interpreting that as coupling reduced in older age, but it could be comparable to less different across the two trial types, right?
Yeah, that's right. So that's a good question. We broke this up also to see if this is driven by the negative or the neutral, because it's difficult with the subtraction to know, but it was a reduced inverse coupling for the negative in this. I should probably just show that. It's kind of hard to have this double difference.
Okay, you're saying that the interpretation is reflected in.
Yes, exactly. Yes. Okay, so wrap it up a bit. Okay, sure, I can do that. So I want to get back to the historical minoritized groups that we're interested in, because it's not a surprise, probably people in this room, but non-Hispanic white adults are overrepresented in studies of cognitive aging in all studies, but all studies as well. And we know from some work that depressive symptoms may be more somatic than affective, leading to under recognition in these historical minoritized groups. I'll skip over some of these things, but we think there's under recognition of depression in these groups for a lot of reasons. It could be cultural stigma, could be lack of access to medical, health care, mental health care too. And so What we don't know is whether depression carries the same cognitive risk in these minoritized groups, but there is some evidence to suggest that it's even greater. So depression in Black and Hispanic adults, when it's observed, when people are looking for treatment, does seem to be related to worse cognitive performance than non-Hispanic white adults, especially in older age. And so in a new project, we are examining this sort of double jeopardy effect in Black and Mexican American participants in a multi-site study called Mind the Next Dog. So these are some of the questions that we're interested in. So what extent do depressive symptoms and age interact to impact cognition and brain health in these historically minoritized groups? And what are the underlying mechanisms of these depression-associated populations? I am going to skip a couple of things and just kind of give you this overview of what the project is. So this is a multi-site R01 where we're, I was going to say here, UT Austin and Georgia State recruiting Black, Mexican, and non-Hispanic white adults. They have multiple visits. They come in for this clinical interview, which is like a depression interview. This kid is a history of depressive symptoms. That helps us better understand whether this is like depression across the lifespan that this person is experiencing or late life depression, which may have different underlying symptoms actually. We're assessing doing cognitive battery, measure any social, structural health, education quality, race-related stress, religiosity, social support, etc. It's a multimodal imaging project. We have multiple memory tasks because we're because we're manipulating demands on executive function within these episodic tests of executive dysfunction. And for people who are over the age of 65 and older, they come in 2 years later for a follow-up visit to assess potential cognitive decline as a function of baseline levels of depression. We can also assess the bidirectional. So that is essentially it. This is just kind of an overview of how I think about all of this work. We're interested in these factors because they are potential, right? We can potentially do something about habitual sleep. So dabbled and probably continue dabbling in some mindfulness intervention, which is for sleep. There's also sleep hygiene interventions, mood and regulation. Now, depression is not, right? If it was so easy to treat depression, there would be no depression. And it's a huge problem. But there are treatments. There are emotion regulation strategies as well. So vascular health is another modifiable factor that we think is actually probably even more important, more of an important predictor of depression and cognitive impairment in Black and Mexican participants. I didn't show it, but we have some data that suggests that. Right? We're also interested in all of these social structural determinants of health that I kind of talked about. I didn't talk about acculturation, but in our Mexican American participants in particular, we're measuring the acculturation to this host culture and how associated acculturative stress may be impacting function. Associated. So I'll end there. We'll end here. Thank you all and all of my collaborators. Thanks.
Yes, I'll ask a question. It's probably on a lot of people's minds, and I'm sure it's on yours, which is the huge gap between associations and causal mechanisms.
Yeah.
So, for example, in the case, say, of depression, one doesn't know straight off the bat the relationship is, for example, between depressive symptoms and then MCI, potential Alzheimer's. It could be the same brain changes which are going to ultimately lead to Alzheimer's, so leading to emotional dysregulation. So how do you get these observations? Same is true, sleep and Alzheimer's that's just a horrendous argument as to what relationships. How do you see yourself getting from the observational data to actually a causal model which actually allows for informed intervention?
So I think that's a great question. My honest feeling about this is that to really assess causality, we would need much more data. We would need data for probably a long period of a person's life. Not the best that we can do, and the best that people can do in literature, is ask, right? So we have a clinical interview, ask people about current levels of depression, past history of depression. This is for depression. We also ask about sleep. And we can measure depression at baseline. We can measure depression 2 years later in this 5-year study. And we can perform, cross-lagged analyses to assess by directionality whether depression is a stronger predictor of cognition than. But I think that this is a difficult question to answer.
Why not do a clinical trial where you take a bunch of people
Sure.
And some of them treated
Yeah.
with depression, some are not, and see what the outcome is 3 years later. So
There are people who do that, and there is data to support that treatment does improve cognitive health. Most of those have not, but that does suggest that depression can be ameliorated and some measure of. But I think, in older age, if there is pathology that can underlie depression and also sort of prodromal evidence of cognitive impairment, it becomes very muddy to assess it. It probably is bidirectional. That's probably true. I think the best that we have is evidence from younger, where like worsening sleep associated with worsening. That to me suggests some. I think it becomes honest.
What do you mean by are you talking about the presence of proteins in the brain?
yeah, so if you think that depression,
You asked about Alzheimer's, right?
yes, if you think depression is actually causally linked to development of Alzheimer's disease, then you would want to like some kind of trial and ask whether that has consequences for accumulation of amyloid, tau, down the line.
Because I think the problem is a lot of the depression medication
Yeah.
isn't necessarily very important. Iterate options.
This is a thought experiment as well.
Yeah, but I guess I thought what you were going to ask is there data, epidemiological data, that should demonstrate dedicated risk of depression or particular SSRI or particular medication is associated with decreased or increased incidence of Alzheimer's disease.
So that's a good question. Not that I'm aware of, but there is data on a number of in terms of cognitive outcomes, but not Alzheimer's disease. And that could partly be due to the fact that these medications were not designed for older and are being prescribed. I mean, a lot of older people take antidepressants, but that hasn't always been. So I don't think that there's the history that we have with younger people to take advantage of for a long period and measure cognitive and remission of depression. We just don't have a lot of that data in but so I think just the answer to this question as to whether these medications themselves are somehow modifying pathology.
And then in terms of, there's issues with conceptualization of modifiable factors linked to dementia, but one of them is social isolation. Do you think the process by which depression might work on dementia risk is via social isolation, and could that be exacerbated in, particularly in Mexican Americans, if there's language barriers prohibiting interaction?
Absolutely. So that is a very interesting question. So we have in our interview some questions about that. It's difficult, but that could be. So if you are experiencing depression or body pain, whatever the symptoms are of depression you're experiencing, that could lead to less physical activity, that could lead to less social engagement, which could exacerbate, right? Poor cognition. Something else about that.
I mean, there's issues with food, well,
Like food choices, and that's
or just alcohol habits.
yeah. So there could be, it's like mediators and mediators.
But all these projects study one thing, so that's the problem, right? Hardly any studies do like, triple, quadruple effects. It's all synergistic, studying it one by one. Depression and sleep, it's got to go in the same study, right?
Oh, yeah.
Yeah,
Yeah.
so, and that's why it's like, well, what's the answer to this, or what's the answer to that? You get a different answer every time, so you have different samples who are with different problems, right? And so it has to be synergistically tested, which means you get a couple hundred people in the sample.
Yeah.
Yeah.
Yeah. I mean, I think that this is going to be a very unique data set because we're matching level of depression across these racial and ethnic groups, which is not easy. Do have a wealth of data, and it's huge for cognitive neuroscience. It's small in terms of things, but those other big studies will get tens of thousands of people who don't have the deep dives. But many students have. Anyone on the spot?
So we have 5 questions online.
Oh, do I need to do it?
No, I can read them to you. So the first question is the following. Do work-related or recreation-related factors other than religiosity affect or attenuate the race-related stress impact on sleep, or were no other factors examined?
Work related? Are there other kind
Yes.
of protective factors? So we looked at social network size, and we've also looked at quality of social networks. So that people are asked about how many close, people in their lives that they have that they could share things with, and that is also predictive, particularly in the Black participants in that study, if I'm remembering the results correctly. So that's another factor, which is not unrelated to religiosity, but they contribute unique variance to the outcome. Did I answer that question? No. Okay. What were the other? I feel like I'm
So the other question is, are there socioeconomic causal differences in anxiety-related sleep losses?
100%, yes. So that is one of the structural determinants that we're interested in measuring. But there is also data showing income-related disparities in sleep quality, and part of that could be because of the environments in which people are living. So a noisier, more densely populated environment is associated with worse, sleep. So that could be part of it. Yeah.
Thank you. So the next question is, if you believe that poor sleep leads to higher incidence of relying on familiarity, what would be your ideal design to determine if improved sleep quality and stability truly improves recollection of items and is not due to other features within those exhibiting familiarity-based reliance?
Okay. Wait, what? I need to hear this one. Remember, I woke up at 1 a.m. So if I
I'll read it again. No worries. It says, if you believe that poor sleep leads to higher incidence of relying on familiarity, what would be your ideal design to determine if improved sleep quality and stability truly improves recollection of items and is not due to other features within those exhibiting familiarity-based reliance?
Okay. Well, I guess what I would do this is a thought experiment. If I can improve sleep quality in the same people, then I would design the experiment that differently and just directly assess familiarity and recollection, which we didn't in that study, to be fair. And then see if it changes the, pattern of responding and also neural activity associated with, or neural indices of recollection. So that would be how I would test it. Think about, if it's within the same participants and they improve, that would be better than different participants for excluding other factors. But I guess I need to think about what the other factors that underlie this response.
Thank you. So the next question is, have you studied whether oral supplements with a natural N-methyl deaspirate antagonist and GABA-A agonist magnesium can improve sleep duration?
No. Great.
Okay, and the final question is the following, or questions, I guess. How do you feel that positive valence items would be affected by these emotion regulation processes in older adults? There's already a positivity bias in older adults. Do you hypothesize that part of this may be modulated by depressive symptoms? Did you include positive items in your evaluation?
That's an excellent question. And in our new study, we have positive stimuli. And I will say from my own lab, we have never shown a positivity bias, never shown a positivity preference. We've only shown reduced negativity preferences in older people with low levels of depression. So I don't know how, that's a longer conversation, but I think that absolutely those studies, this idea that there's this positivity bias with increasing age and this, socio-emotional selectivity theory, I think it's perhaps right for a select group of older people who are high income, non-Hispanic white, and not depressed.
Not NIH supported.
And not support. So I think when you start to increase the generalizability of your sample and diversity of your sample, then you may see different effects. And that's what we've always seen.